This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 291, recorded on July 6, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
I bet it's 90 degrees there, right?
It is indeed. It is indeed. And the humidity, it feels like we're still in Houston.
That was something. Also joining us from, I don't know, is it Ann Arbor or Washington,
Ann
D.C.?
Arbor, Michigan.
Michelle Swanson.
Hello, everyone. It's very pleasant here. We've got a nice breeze, bright sunny skies, 81 degrees Fahrenheit. I'm happy.
And from St. Louis, Missouri, Petra Levin.
It's lovely to be here. Our weather is decent today. It's sunny and a little breezy.
And returning for the nth time, our TWiM adjacent Mark Martin. Welcome back.
It's nice to be here, friends.
Hey, look at Mike. You got him to hold the mic like a rock star.
I did indeed. Ray will be happy.
Many things. That is not one of them.
When we do the live TWiM and TWiV at ASM Microbe, we have to hold microphones. And Ray makes sure he gives us instructions. Hold it like an ice cream cone. Works very well. They're good microphones. Anyway, we have a interesting mix of science for you today. And we have not yet released the episode from ASM Microbe. That should come out next week. And, of course, me saying that means nothing because this episode will come out after that one. So
that's true.
It's the space-time continuum, you know? But when you hear me saying these words, you will have heard that Mimi Goldschmidt. And hopefully you're still laughing because that was a great episode, right, Michael? And
it was. Mimi was her old self. She entertained the audience and gave them some of her many years of wisdom.
And Mark, you were in the audience, I believe, correct?
I was.
Yes.
And it was fabulous. I have lots of relatives, and every Texan will not be happy with this, but my mother and my grandmother were from Oklahoma. So when I hear traces of that when Mimi was speaking, and so it made me feel at home.
Excellent. All right.
Delightful.
So we're going to start with a snippet from Mark. If you thought bacteria were great, wait till you hear this. Oh my gosh. Go ahead, Mark.
Well, I'm very delighted to be here and talk about all this. This is fabulous stuff. The first thing I wanted to say, it was great to see everybody at Microbe. I miss seeing Michelle because Michelle is usually a high point of my visit. But I did get, it's true, but I did get a plush immune cell from Petra, who just walked by and handed it to me. That was pretty fabulous.
Which kind of immune cell is it, Mark? Do you know?
It is a type of a B cell because it was producing immunoglobulins.
Oh, very good.
Yeah. So, I don't care for many eukaryotes, but that was a good one to have.
What do you mean you are a eukaryote? How can you not care for them?
You like us.
Children. And
you
you
like
have
us,
children.
Yes. Well, I'm a holobiont. You know that. So, but anyway, you're right. There are some eukaryotes of which I am fond. So I wanted to keep this short as a snippet and be a true snippet. And this is because of the tangy, super sweet toothpaper that Michael's going to talk about. I like the acid and the sweet on that. But I also...
and they're both, if I can give our listeners a heads up, they're both about biogeography that affect tectonics and teeth.
Oh, good title.
We have an alliteration.
Yes.
Michelle, I have missed you. That is so fabulous.
And it's microbiogeography, as a matter of fact.
And they're both open access, so you all can actually follow along with us.
And just parenthetically, before I get started with this, you're going to look at the actual journal that this is in. And yes, Michelle, I will give the authors' names and everything this time. It's Geophysical Research Letters. And, you might think, well, what can a microbiologist get out of that? And the answer is plenty. I'm really interested what you all think about this. But again, I want to be very conscious about time. And a couple of things before I get started is that as an educator, I find that most of my students are primarily interested in human-associated microbes. I call those hashtag devil microbes or hashtag angel microbes. And mostly microbes are what I'm going to call hashtag meh microbes. They could care less about us. And it's really true. Now, ecologists I know often teach about how microbes cycle elements. But again, for the students and for a lot of professionals, it's the medical emphasis that seems to predominate. But I would suggest that microbes, to borrow from Beyonce, rule the world. And one of the things that I push my students towards seeing a lot is what I call deadly centrisms. And one of them is oxycentrism, because we're so used to harvesting electrons from NADH and dumping them on oxygen. But that's not how the majority of the world has been. So if I can borrow from the Dean Martin story about Frank Sinatra with you, it is a microbial world. We just live in it. And so this paper, Photoferrotrophic Bacteria Initiated Plate Tectonics in the Neo-Archean, really caught my eye, as you might imagine. Now, Cameron Thrash on Twitter, we'll see what happens with Twitter, is a great follow because every day he posts papers that I'd never heard of, and this is one of them. So I really recommend people do that. And Cameron, thank you. It's, as I mentioned, Geophysical Research Letters, May 2023. And the authors are Zhang, Li, Leng, and Gurnis, various institutes in China, and also the last author at Caltech.
Mark, thank you for giving a shout out to your colleague on Twitter, because I was wondering, I'm so impressed by your breadth of knowledge, but I'm like, wow, how did Mark find this paper?
I should just say that I was just casually reading around, but it's not true. I listen. Hard to believe, but I listen. So in any event, the first thing about the paper is, again, I'm worried that some people might look at this and say, I'm not a geologist. I don't want to know more about it. First off, this is a great paper. There's some stuff in it that's hard for my innumerate mind to carry, but there's so much good biology in it. And I adored the key points that you see on the left-hand side of the paper itself, the clear abstract, and what amuses me no end, the plain language summary. I like that so much.
Which is not so plain, actually. Right.
True.
In the second sentence, they go subduction, which not my plain
I looked
language.
that one up. I looked it up.
I remembered that from like earth science in 8th grade. Or something.
Well, you listened.
Yeah.
I didn't. That's the problem.
And I don't know what mechanical calculations are.
Oh, well.
It's math.
Well, I know.
Math. It's math. Yep.
May I quote you elsewhere in my household today, Michelle?
Yeah.
Say no.
Well,
Say no.
Oh, yeah.
So anyway, the idea that microbes can change the planet's been around for many, many years. I remember Lovelock and Margulis's Gaia hypothesis, and a lot of folks didn't take that seriously, that all living things on the planet are a superorganism that regulate and alter things themselves. It's absolutely true that there have been several mass extinctions on the planet due to biological activity. The Great Oxygenation Event is one of them. And if you haven't read Peter Ward's The Medea Hypothesis, I strongly recommend it. I put links up for this. There's even a Scientific American blog post about Dr. Ward's work. So that idea was fine with me. And I thought, well, lots of things are going on. And I collect fossils and rocks and things of that nature. And you're looking at a banded iron formation sample here.
That's beautiful.
If you polish it, it's even more beautiful. Yeah.
Is it magnetic?
I actually haven't tested that, which is interesting. I don't know that it is based on the crystal structure. Certainly the red that you're seeing is either hematite or magnetite, but I don't know it's an oriented crystalline structure. But I love that idea.
So because we're a podcast, let me just hold that specimen up. It's a
Certainly.
beautiful slab of rock, but it's got beautiful streaks of different colors running through it in a very ordered pattern.
It's got red bands and gray bands and some yellow, which
some yellow too.
I'm not used to seeing. And banded iron is actually very famous. I know this is a snippet, but banded iron is the cause of much debate on Earth, on origins of life on Earth and oxygen and where do you get iron oxide and when did it come? There's a lot of debate around, and it all goes back to the banded iron formations.
Let's talk about those.
Let's do that. And early Earth, we can agree, was a very strange place. And I love handing out things like this when I teach about it, because students tend to glaze over a little bit, and they've got a lot on their plates. I'm not unhappy with them when I talk about the geology of it. Now, ferrous iron, Fe2+, is more soluble than ferric, Fe3+. So ferrous iron is quickly converted to the ferric form by oxygen. So currently, we don't have a lot of ferrous type iron in the ocean because it has precipitated out in the ferric form. So that's one of the things, the great oxygenation event had happened. And there were cycles that happened. So you get the layers that Michelle and Petra were talking about of some type of iron-bearing mineral, and usually layers of chert, a condensed quartz-like substance, okay? So there were clearly lots of cycles that went on this way until the cyanobacteria had their way with us. And the early ocean was highly anoxic, and there was not yet a lot of sulfide. This is the interesting thing that I tell students, is that when you take an electron from NADH and you dump out an oxygen, and you have this huge energy differential, and that's what drives what we know about in metabolism. There are other ways to do those things with different donors and acceptors, and it can turn into a laundry list very easily. But the take-home message is many types of microbes to this day can use inorganic compounds to do that. For example, ferrous iron in solution can donate an electron to biological systems, thus becoming insoluble ferric. And that really brings to mind what Norm Pace always said, that metabolism is all about the electrons. Again, when we talk about respiration, the idea of oxycentrism, just like eukaryocentrism, is always with us. Now, all of that's well and good. We all have a laundry list of different types of things that could happen. But what is a photoferrotroph? A photoferrotroph is a type of an organism. Now, they used bacteria in the title, and I'm a little nervous about that, and I'll tell you why later. There certainly are bacteria doing this today. What they're able to use is ferrous iron as a source of electrons. They use light, and then they're actually able to fix carbon. And generally speaking, most of the green sulfur bacteria today, some of the purple non-sulfurs, and most of them are using reverse TCA to bring in carbon from carbon dioxide instead of our good friend Rubisco. So all of that's
Calvin cycle, or the Calvin cycle.
I'm really glad to hear you say that. Exactly. Rubisco is simply the first step, the fixation step, and then you have reduction and regeneration. In any event, plate tectonics, we were talking a little bit about that earlier. And it's the idea that you have floating islands of rock on top of the magma, essentially, and they're abutting one another and pushing at one another. And this is the source of some of the earthquakes that I personally experienced. And when you have two plates close to one another, one tends to go under the first one, and that's called a subduction event. So if you think about it, that keeps bringing oxidized material down to the interior, and then that brings it back up again. So it's this wonderful, I hesitate to use it like a bubbling pot. That's not exactly it. But I think you know what I mean. But the real question is, how did plate tectonics start? And the paper does a really good job talking about that and giving different hypotheses for it. I noticed there's a little bit of, I'm not going to say lack of clarity. I think it has to do with different ideas having to do when plate tectonics happened. It certainly
started. We know
when it
plate tectonics. We know it's happening, but it's really about the chicken and the egg part.
The initiation event.
Yes.
Right. And as Vincent knows, when it comes to the chicken and the egg, which came first, it was the retrovirus.
That's right.
And he knows that to be true. And you know what I mean, Vincent.
I do.
So in any event, how this happened, they've talked about lots of different things having to do with how magma circulates or even asteroid impact. And so I'm reading this thinking, okay, tell me more about the bacteria, because that's what I'm thinking about. But the critical idea is the weakening or even shattering of the lithosphere, which is the layer of rock sitting atop the magma. And then you have the beginning of these subduction zones, which are really important. Cycling of elements, energy, all the rest. The authors are proposing that banded iron formations can be dense enough to fracture the lithosphere over time and beginning those subduction zones, beginning plate tectonics. Banded iron formations can be 40% denser than the lithosphere. So this is a possibility, they say. And I draw your attention to figure 1, which I've heard a lot of stories over the years about microbes that breathe metals, for lack of a better term. But I have never heard about iron deposit as being fluffy.
Yeah.
Mm.
And I would argue it is correct to say that when you compare hematite, which is fluffy, not to you and me, but to geologists, to magnetite, which is quite dense. And what's interesting is they show in figure 1 what happens at these protocontinental margins and driven by microbes and hydrothermal vents. So what they're suggesting is in the photic zones around there, you have a fair amount of ferrous iron in the water, and the photoferrotrophs are using it, and they produce ferrihydrite. And that compound reacts with more ferrous iron, which comes from nearby hydrothermal vents, and that causes the precipitation of magnetite. They have some lovely numbers talking about the difference in grain size and density between hematite and magnetite. And I should point out that after the great oxygenation event, the product you mostly get is hematite. Before that, it's mostly magnetite. So, you know what Sherlock Holmes said is that after you eliminate the impossible, whatever remains, however improbable, must be the truth. And I think they're onto something here. We know when these BIF-associated basins formed, and they're near the time of the beginning of subduction zones, it's estimated. And those photic zones are near the edges of those protocontinental shelves. And the favorite parts, I believe, on page 3, where they talk about a 50-meter deep photic zone with a cell density of about 5 times 10 to the 4th cells per mil, should be sufficient to drive that precipitation of magnetite as described. And a few million years, and I love the cavalier few million years.
Yeah, only 9 million.
Yeah, right?
Well, I like the 1 kilometer per million years of deposition.
Yes, 1 kilometer. And the other thing, Mark, that these authors may not have appreciated, but in the magnetotactic bacteria, the form of magnet is magnetite, the same element that they are cooking up here. So I found that, and I was surprised not to see that somewhere in their discussion, since they're talking about magnetism and sinking, because the working hypothesis surrounding magnetotactic bacteria is it drives them deeper into the sediment. So the question is, are the magnetite deposits independent of the photosynthetic event actually dead magnetotactic bacteria?
They form single domain crystals, Michael.
I do.
And in fact, I went to New Zealand a few years ago. It's one of my peak experiences. And there's a beach in the northern part of the North Island, which is called Magnetic Sand Beach, because, Michael, it's nothing but magnetite. That's the sand.
Yes.
And so I actually asked if that was the product of some kind of biological action. And I was told, oh, no, that's volcanic. I don't believe that. I believe in what you just, because you don't see.
This paper is effectively supporting your hypothesis, Mark, that it's biological in nature. And it's really quite remarkable that if you can get a volcano that's spewing out iron as an energy source for the microbe, and they have a photosystem that can effectively get them the electrons they need to fix carbon, it really is a pretty neat science fiction story, so to speak.
I think it's science. I mean,
it is
a really compelling argument. They're drawing many different fields to build a really coherent case. And those volcanoes that are spewing the iron, they're at this period in our globe's history, they're under the water. So they would be spewing it near the surface where these bacteria that are using the sun and iron to metabolize reside.
It's recycling.
So it's all very plausible to this microbiologist.
Mark, are there any holes in this story?
I'm sure. Basically, well, no. I think that when you say that you get these dense banded iron formations capable of fracturing the lithosphere and beginning this at about 2.75 to 2.4 billion years ago, sure. When you start digging in to ask when plate tectonics happened, or began, as Michael says, it kind of reminds me of when students ask me, did viruses evolve from simplified cells or complicated viruses or from transposable elements? And I say yes.
Yeah, that's a yes. Absolutely.
Right. In a similar way, there could be multiple things going on, but it is hard to ignore the density arguments. And we know that those BIF deposits, those banded iron formations composed of magnetite, are there. They're the product of biological activity, and they are heavier than the lithosphere and the mantle.
Yep.
So I find it very hard to believe it.
A lovely figure 4 where they break down history, ancient history, and then the different events, and show how it all kind of times up.
I think it plausible. I'm a little hesitant, to be honest with you, Michelle, because, and I think Petra alluded to this, there's some controversy about the timeline of the oxygenation event. And people get really unhappy. They get agitated. So I dislike people getting unhappy. So I'm willing to accept more than one thing at once.
And the oxidation event would have brought the end to the deposit of these heavy iron deposits that would have pushed down the plates and caused these big shifts.
Well,
because it's not so fluffy.
Well, the limitation in that is we don't really know when sulfide became the limiting reagent in the atmosphere for the other microbes to use it as a source of electrons to effectively make more microbes.
This is, I mean, all of this is like, but in the same way with banded iron and, the big oxygenation events, the question, I mean, how do you know what you know? I mean, they're basically just trying to piece together across geologic time, where 9 million years is not very much, actually, right?
Correct.
It's like nothing. And, the weight on the idea that you have something so heavy it pushes through is also just hard to wrap my head around. We have things that sit on ground, and as long as it's solid, it doesn't go anywhere. So, the compaction, I mean, they're basically having to make a lot of assumptions. And then, but it's a really interesting idea because, you're taught this is like earth science, physics, not related at all to microbes. But maybe microbes are doing even more than I like to think they're doing, which is everything. So.
Well, and the authors are using computational models where they take the data that we do have and build it into models and then ask, does the model generate plausible answers?
It's like it's a form of biophysics. Assume a round cow. So assume a round earth.
Right, a round cow. I just read about that in physics, yes.
So There was a lovely short science fiction story that I know that Michael will enjoy hearing about. It's called The Green Marauder by Larry Niven. And you have aliens who've been traveling near the speed of light. So they were visiting Earth 1 billion years ago. And they're telling these other humans that there used to be a life form here. It was very different than us, and it was slow. And they started to have trouble because of this slime that was appearing near their cities that was giving off this poison. And of course, what that means is the cyanobacterial revolution, right? And that idea is something that is just amazing to me. The idea that you can have a biological activity that destroys almost everything on the planet. And it's a product of biology. And you'll find, if you read Peter Ward's book in particular, lots of examples of this, the relationship to snowball Earth, the great dying, which might be a matter of sulfide, by the way, Michael. It's really interesting stuff.
Well, I think the most telling line or most shocking line to me in the whole paper was their model implies that prosperous photoferrotrophy in the Neo-Archean is the reason why Earth is the only planet with plate tectonics.
The only known planet. Maybe they should.
I would never say
that.
Vincent and I sat next to each other when we heard Andy Weir talk at Microbe. And Andy Weir, of course, is the author of Project Hail Mary, The Martian, among other books. And he says he just makes stuff up. And, as long as it's plausible. And, this is, the math is interesting, but I was just like shocked that you need a biological input in order to literally move mountains.
Yeah.
Because that's what platonics does. I mean, that's after all why we have Mount Everest. It's why India is slamming into the Himalayas and all of that.
But that's just to start it. But then what maintains it?
What keeps it going?
What keeps it going?
Good question. I was going to ask that. Yep. Does anyone know?
It's supposed to be in fact, there are patterns of magma moving all the time, and there are asynchronous areas. Mm-hmm. There's something under the Pacific that's much more dense than the areas around it. And we know that because satellites actually get a little bit closer as they pass over it. And they're also, they're found in other planets. The moon has them. They're called mascons for mass concentrators. And I think it's lumpy on the inside is what I'm trying to say. And then when you figure that you have this rapidly spinning electrically conductive material inside the Earth, that might have gotten the whirligig going. But I will be very interested to see whether or not we know there's nothing like plate tectonics on Venus. We know that. Mars, I don't believe so either, but it doesn't appear to have a spinning core either.
No, they lost it because that's why they don't have the shield, the magnetic shield.
I am curious about what this biogeography has to do with cavities.
Here we go.
And not the Marianas Trench kind of cavity.
No.
Thank you, Mark.
You're very welcome.
It was a fascinating story. And I'm going to take us into a completely different direction, but we're still going to be making something. The title of this paper that appeared in Nature Communications is also an open access paper, is Selenomonas butygenia acts as a pathobinont, mediating spatial structure and biofilm virulence in early childhood caries. And it was published on May 22nd. There are 16 authors, and the corresponding authors are Devarius, Wu, and Ku, and they are principally in 3 departments. One is at the Department of Biostatistics at the Gilling School of Global Public Health at UNC Chapel Hill. The other is the Biofilm Research Laboratories, the Center for Innovation and Precision Dentistry at the School of Dental Medicine at the University of Pennsylvania. And the other is the Division of Pediatric and Public Health at the Adams School of Dentistry, also at UNC Chapel Hill. And so today's paper is going to address an infection that unfortunately affects 600 million children globally. And no, it's not diarrhea, but rather it's tooth decay. And I think that number of 600 million children globally really tells you that this is a pretty significant infection. In today's paper, we're going to discover how biofilms within our oral cavity can impact the virulence profile that then manifests itself as early childhood caries or the decay of teeth. Now, we've all been to our dentist, and each of us has secretly hoped for that clean report that offers us that glorious news of no cavities, no gingivitis, no periodontal disease, and it's not yet time to address our gum recession, something more dreaded to hear than hairline recession in men. And so, and if you've ever had gum surgery, I'm sorry, it's really quite an ordeal, and it helps treat the periodontal disease, but it is quite an ordeal. Now, this paper has a bit of everything. It has a clinical trial. It has a mechanistic discussion, a mechanistic proof, some hardcore microbiome analysis, and some of the coolest images and videos supporting the evidence as to how teeth decay that I think I've ever seen up close and personal. And it gives us a new meaning to the term cage match, for that is precisely what the microbe Selenomonas offers to enhancing the virulence of strep mutans in its ability to form a dental cavity.
You talking from experience, Michael?
No, I haven't had gum surgery. No,
I'm talking about cage matches.
Oh, no. Cage matches? Well, I guess you could say the closest thing any of us have ever had to a cage match was our qualifying exam. So many graduate students refer to it as that.
So, Michael, you're an expert in dental microbiology. Is this species name Sputagina? Is that like Latin for spit?
Yes, it is indeed, Michelle.
All right.
So we could just say Selenomonas spit, but that has an inelegant form to it. So as background, what is the infection commonly referred to as dental caries? Well, our teeth are part of our digestive system. So it only makes sense that our intestinal microbiome would be associated with their care and maintenance. Most of us have 32 teeth. While some of us may have more, some of us may have fewer. I actually only have 25 teeth. The orthodontist got 4, and then the oral surgeon got 3 more. So enamel is that protective layer that covers the crystal lattice that we recognize as our tooth. And while our teeth might look like bone, they are actually an ectodermal organ. And other ectodermal organs include your hair, your skin, your sweat glands. And so those of you who have taken developmental biology know what beget what. They're literally the cells arise from the ectoderm that make our teeth. Now, the enamel helps shield our teeth from the cavity-causing bacteria, which is the topic of this paper. And dental enamel is the hardest and most mineralized tissue in both extinct and extant vertebrate species, and provides maximum durability that allows our teeth to function from everything as weapons to tools, and of course for food processing. And enamel development and mineralization is an intricate, tightly regulated process where the cells of the enamel organ, called the ameloblasts, are heavily polarized, and they secrete a monolayer around the developing enamel tissue and move as a single forming front in a specified direction as they lay down this proteinaceous matrix that to many of us really appears to be nothing more than clear nail polish, but it serves as this template for crystal growth that we know as our tooth. And dental caries occurs when the biofilm that normally resides in an oral cavity in a homeostatic interaction changes from relatively a neutral producing biofilm to one that generates acid. They become acidogenic or acidoduric, where they can really, the biofilm can really tolerate the lower pH, often 4s and 5s in pH, that then drives the dissolution or drives the disequilibrium of that ectodermic tissue that is our tooth, that is principally comprised of a remarkable crystal lattice called hydroxyapatite, which is an inorganic mineral that's assembled into a lattice of 10 parts part A, 6 parts B, and 2 parts C, where A is calcium, B is phosphate, and C is hydroxyl ion. Now, pure hydroxyapatite contains about 39.68% by weight of calcium, 18% of phosphorus, and it results in a calcium phosphorus ratio of 1.67. And when we lower the pH, we change that ratio so that the calcium that was normally in saturation, so it is a crystal at that point, comes back into solution. And that's effectively how you get the beginning of the caries formation. So here's the story in today's paper. Fermentable carbohydrates are metabolized by the biofilm containing bacteria that in turn produce organic acids, primarily lactic acid. And these end products of bacterial metabolism then accumulate in the fluid phase of the biofilm, causing a pH drop that then drives that disequilibrium on the surface of our tooth. Now, we all hopefully have access to fluoridated water, and the substitution of the fluoride ion for the hydroxyl ion actually helps to change that dynamic equilibrium constant, resulting in a more stable or acid-resistant crystal lattice. For the fluorine swaps out for the hydroxyl, changes the disequilibrium constant associated with calcium, and that's why we put fluoride in water. With that as background.
Hey, Michael, can I ask a question?
Sure.
It's my impression, correct me if I'm wrong, caries are mainly a kid thing, adults less frequent, right?
Adults can still get caries, especially in populations that have dysbiosis of the oral microbiome. Principally as a consequence of diabetes is a good one, because you have a higher sugar content, and you can have smoking leads to inflammation, which then brings in other microbes that add to this dynamic equilibrium of dissolution. It's not only strep mutans. It's not a pure culture infection. There are many organisms involved, and that's what we're going to learn in today's paper.
Is it the case that children more often get caries than adults?
It is.
Okay, thank you.
And that's simply because of the issue with the enamel, and they're evolutionarily designed to be deciduous. We lose our primary teeth.
So they're getting those cavities on their baby teeth?
Yes.
Not on their adult teeth?
No.
Okay.
But if you have a large proportion of these cariogenic biofilm organisms, as you begin to lose your primary teeth, then they will form the basis of the biofilm that associates with your permanent teeth. It's a founder effect, something that drives ecologists crazy and makes the world go round. You know, who gets there first?
So they should not treat their baby teeth as loners.
No, they should not. And in fact, that's why many children, when they go to the dentist, they offer to put sealants, effectively a layer of nail polish, on top of the enamel, so that if they already have the bad biofilm, they can actually slow things down. And then through normal growth and the normal changes in hormones, because hormones can change it. Pregnant women have a different oral cavity, and I'm going off topic. So let me go back to the
before
paper.
I got another question. I'm sorry. So why don't we treat caries with antimicrobials?
There's too many of the suckers in there, and it's sort of like using a nuclear
explanation.
Well, it's sort of like using a nuclear weapon to take out the gangs in Chicago.
And in general, biofilms are hard to clear with antibiotics because they're not permeable.
Slow. Okay. They're growing slowly. They're not permeable.
I know listeners are going to ask that. That's why.
Oh, yes, they will indeed. Which is why I went into the background of how caries form.
Yeah, but there's so much beautiful data in this paper. We better get rocking.
We got to get rocking. All right. So strep mutans is a homofermentative gram-positive biofilm promoting pathogen. It's been implicated as the primary pathogen in childhood caries. While the role of polymicrobial communities is appreciated, it remains unclear whether other microorganisms are active contributors that interact with the primary pathogen, which is how this microbe, Selenomonas, comes into the picture. What they're going to present to us is how they integrated a multiomics supragingival. That means the organisms below your gums. So these aren't the ones that are stuck up in there that you're pulling out with dental floss. These are below the gum line. From 416 preschool-age children, 208 males, 208 females, in a discovery validation pipeline to identify disease-relevant interspecies interactions. And then they demonstrated what they were proposing. What they learned is they found 16 taxa that were associated with childhood caries from their metagenomics and underlying this word, metatranscriptomics analysis. So this paper goes beyond just doing a census of who's there. They're going to tell us through their transcriptomics that's going to give us insight into what mischief the microbes are up to in this biofilm community. Then they took us through a multi-scale computational imaging and then virulence assay, where they examined the biofilm formation dynamics, the arrangement of the biofilm, who was where, how big they were, what they were doing with each other, and then the metabolic activity of the oral flora microbes. And Selenomonas, which is an anaerobic gram-negative bacterium that is generally associated with upper respiratory tracts of people. It's not necessarily associated with caries. And in point of fact, Selenomonas can't cause caries by itself. It needs something else. And Selenomonas is generally associated with gum disease. And if you've ever had a root canal, it's associated with endodontic infections, which are an infection of your root. But by itself, Selenomonas cannot damage enamel or that protective layer. And then there were three others that they really focused on. Prevotella, salivae, which is another polite word of saliva, and then Leptotrichia waddii, which is a gram-negative anaerobic non-spore-forming pencil-thin shaped bacterium that is part of the normal oral and intestinal flora. And they typically occur in our oral and vaginal cavities. Now, they offer that Selenomonas, this flagellated anaerobe, which is motile, with a previously unknown role in this supragingival biofilm, becomes trapped in the streptococcal exoglucans, then loses its motility, but then actively proliferates to build a honeycomb-like multicellular superstructure that encapsulates the strep mutans in a cage, enhancing its ability to concentrate that acid in one spot of our tooth. The images here are wild. Like I offered in the opening, they are creating a cage for the cage match between strep mutans and our dental enamel. And then their movie actually really illustrates that pretty well. And if that's not going to be enough for you, go ahead, Michelle.
Can I jump in? The term trap and cage implies it's a negative interaction.
No,
and it sure seems like these bacteria that are forming these clusters and cohabitating with strep mutans are thriving. They're actually
it's
pretty
mutualism.
happy. Yeah.
This is mutualism. It's bad. It's a negative for the child with this cage match strapped to its tooth.
Michael?
Yes, Mark.
Do you remember the Della Vibrio and Vibrio interactions paper that we did a while back?
I do, Mark.
And how there was that emergent property of new structures together that you don't see individually. Right. That's what this reminds me of. It's beautiful.
Yep.
Yeah, it's the final piece of this
biogeography.
Puzzle, yes. If that's not going to be enough for you, then they're going to offer us data developed from a rodent caries model, which allowed them to uncover an unrecognized ability attributed to Selenomonas to colonize the supragingival tooth surface, like Mark said, similar to what we saw with Della Vibrio, and this remarkable observation that in concert with strep mutans, the Selenomonas was able to cause extensive tooth enamel lesions and was able to exacerbate disease severity in this rodent caries model in vivo. Reducing this all to one sentence, they uncovered a pathobiont that can and does cooperate with a known pathogen to build a unique spatial structure which confers this enhanced ability of an organism that we've known for decades causes caries to foster this significant disease development. Now, as Michelle has already tumbled to, the data they offer in support of this are pretty impressive. Their first figure, though...
Wait, Michael, can you define pathobiont?
I've
never heard that word. So.
It's a microbe that is effectively contributing pathology to the biofilm.
Okay.
So Arturo Casadevall doesn't like that term, but I'll accept it today.
I'm thinking of these guys as they're like focusing the mutant.
It's like a contributory,
like a laser or focusing it. Yeah.
Yeah, it's contributory. I think the best way to look at this is Michelle has already gotten into. It's really mutualism. Both of these organisms are benefiting from this interaction. We only cause it a path because it's causing pathology in the bigger host. But they much like this environment that they have created. The Selenomonas has literally come out of the gum line and invaded a new niche that is quite happy in.
So, Michael, if this was happening on a non-tooth structure, we would say it's a beautiful example of mutualism, wouldn't we?
Yes.
Right?
Yes, we would. If it was happening on a rock that we were talking about earlier.
Well, it's still beautiful. It just, you don't like kids getting cavities, that's all.
No.
But it's still beautiful. I mean, you cannot deny that it's beautiful, right?
It's remarkable.
Yeah.
And their first figure really walks us through how they set up these experiments, why they needed 16 authors. And again, as this is a Nature Communications paper, they describe the role of each author in which part of the paper they played. But it's really, as this is an open access paper, I'm only going to hit the highlights, but to suffice to say that the results revealed there were novel taxa associated with childhood caries. Now, this was not simply...
Can I jump in and
Sure.
just say what was so impressive to me is they started with an unbiased approach with 400 samples from kids and
Clinical
just asked,
samples.
like, who's there, by using whole genome sequencing and then computational biology so they could deduce who is present. And then they also asked, what are they doing, by looking at RNA profiles. And again, a lot of beautiful computational biology to see what enzymes, what proteins, et cetera, are being made. And then based on their knowledge of the biology in the mouth and dental caries, they could then make some testable hypotheses and then shift it into a whole series of in vitro assays to ask, is it consistent with this? Yes or no, yes or no. And then the beautiful ending with in vivo experiments with live animals and showing through really beautiful work that everything they deduced and then tested in vitro is absolutely consistent with what they're seeing in vivo. It's really a remarkable tour de force by these three groups.
And that's effectively how we're going to get through the paper. I mean, they literally lay this out, and the high resolution imagery that they illustrate for us is really something that I think you should open the paper, take a look at, even if you've never looked at confocal biofilm based images, because they're really quite remarkable. And then they used a novel technique that I wasn't familiar with called Mander's overlap coefficients, which is nothing more than a quantitative measure to assess spatial proximity. And they were able to co-locate the microbes in the biofilm, but then were able to show them what actually was going on. And as Michelle said, they were then able to formulate a hypothesis as to what was going on. Bottom lining this incredible imagery, they showed us that the three new candidates form mixed biofilms with strep mutans, and that this coexistence enhanced, so this goes back to the mutualism argument, enhanced their surface colonization and co-development of the structured biofilm, for which Selenomonas formed a densely populated community interspersed between strep mutans. So it's effectively the Selenomonas is the cage, and the strep mutans is in the cage, and it's dumping its acid on.
Or perhaps the term honeycomb that they use is maybe a better one.
Honeycomb is
so.
Honeycomb.
Figure 7A shows beautiful micrographs with fluorescent dyes that distinguish different components, the two different species of bacteria, and also the extra polysaccharide matrix that they use to form these stable structures. And honeycomb also emphasizes it's the metabolism of sugars that kicks this off, and then the acid that builds up, and you get the pathology.
And, that's effectively the whole story. But I would just like to finally encourage you all to review Figure 10, which sums up the approach they used and how they were able to reveal the data they revealed. And it really serves as a roadmap of the value proposition that a discovery validation pipeline offers to provide us new insights into, here in this case, a very old disease, transforming a benign biofilm into a virulent one. And I'm sure that folks like Mark Martin are going to walk their classes through this paper to introduce them to the notion of a multimodal experimentation approach to understand how you form hypotheses, how you combine many methods in order to demonstrate what you think is going on is indeed going on. So I really commend this group. They've done a remarkable service for the dental community in really highlighting how we might then be able to address this problem in 600 million children. So that's it in a nutshell. Michelle, would you like to say something about the authors?
I would. I'd like to highlight the two lead first authors. The first one is Hong-Young Chow. He was a PhD student with Dr. Di Wu at UNC in their Department of Biostatistics in their School of Global Public Health. And he actually started as an accountant in Korea. And I'm sure was doing well, but wanted to do something more creative. So he came to the U.S. to study statistics and biostatistics, and had the opportunity to really get into this kind of whole genome approach. So currently, he's in reinforcement learning research at Amazon, but not before he contributed to this beautiful work. So he was leading the single-cell RNA sequencing and microbiomics approaches, and then was delighted to be able to collaborate with the Devaris lab, who has been studying this children's oral disease and microbiome and really led kind of the clinical part of it. So he was delighted to be able to apply his bioinformatic computational skills on a health problem. So he points out that the collaboration with the Koo lab at Penn was invaluable. They were driving the wet lab experiments, and that allowed them to really test some of the ideas that their bioinformatics generated. So advice to others is to follow your curiosity, know your own skill sets and what others can contribute, and then be persistent. And it took all of those traits to make this paper come to this beautiful report. So as hobbies, he used to do programming as a hobby, but then when he became an accountant, he had to come up with some other hobby. So he started to do marathon running. So currently he's a half marathoner, and he needs the same kind of persistence in this pursuit as with programming. And he says he's not yet persistent to run a full marathon, but it's a goal he's set. So he worked closely with Ji Ren, who is a postdoc in the Hyun Koo lab in the School of Dentistry at UNC, whose path to career in science was really motivated by wanting to apply science to improve clinical outcomes and people's quality of life. So he was actually got a dental degree and a PhD through a joint program between the West China School of Stomatology and Penn's Dental Medicine School under the mentorship of Dr. Hyun Koo, who was a dentist, microbiologist, scientist. So after he got his DMD and PhD, he remained in the lab and became really fascinated by time-lapse live microscopy and generated a lot of the beautiful images and the movies that they published with this work. And then also he helped develop the in vivo rat teeth cavity model. Yeah. And also learned how to work with clinical samples from these early, the pediatric population. He also has worked on the contribution of fungal bacterial cooperation in saliva and has published that in a PNAS paper in 2022, also about biofilms and how they can spread and cause damage. He's currently part of a NIH-funded training program at the Center for Innovation and Precision Dentistry. So this allows colleagues from the Penn's Dental School and also Engineering School to collaborate. So he is really grateful for the amazing colleagues that he's had and the mentorship with people from engineering, from dental medicine, and then the wet lab group at Penn. So he wants to really thank all of his colleagues from whom he learned so much and really made key contributions. A really exciting day in the lab for him as a microscopist was when he, for the first time, caught Selenomonas doing its acrobatics, as he describes. This is a motile bacterium, which is unusual in the mouth. And so he was just blown away by seeing these bacteria jumping around until they got caught in this honeycomb. Mm-hmm. So for junior colleagues, he says, work hard, be kind, exercise, and plan carefully, learn from your mistakes. So, yeah, those are all good lessons.
Wonderful. Thank you, Michelle.
Thank you. Mm-hmm.
Good stuff. All right. That is TWiM 291. You can find the show notes at microbe.tv/TWiM. You can send a question or a comment to twim@microbe.tv. If you like our work, you could consider supporting us. We'd love your support. That's how we make these shows. That's how we have this lovely incubator. Microbe.tv/contribute. Our TWiM adjacent. I'm not going to call you a guest anymore, Mark. You're TWiM adjacent. Mark Martin, University of Puget Sound. Thank you, Mark.
You're very welcome.
What is the white spiky thing behind you on the right? Is that a phage? Yeah.
Oh, this? Yeah. Hold on. And it's got a light inside.
I see. Did you 3D print that? Yeah.
My wife certainly did.
Who we just saw walking behind you, actually.
Yes, you just did.
And who we met in, where were we? Houston.
Houston. Houston. We had a lovely dinner with them.
Very nice. Thank you, Mark. Michelle Swanson's at the University of Michigan in Ann Arbor. Thank you, Michelle.
Thank you. And I want to thank both Michael and Mark for finding these really fascinating papers that show the impact of microbes, whether it's on tectonic plates or dental caries.
Yeah, amazing. We literally span the globe today.
Yeah, but all about the metabolism and the architecture that they form out there in nature.
Yep, it's very cool. Once you may think that we've been wowed as much as we can, and then you get wowed again. Yep. The key is you just need to keep looking.
Mm-hmm. It's all OMG, Vincent.
Overwhelming microbial greatness. I have learned that from you, Mark. Thank you very much. Petra Levin is at Washington University in St. Louis. Thank you, Petra.
Thank you. This is really fun. And I love the biogeography. It's like on all different scales.
Yeah.
Not just, in different places in the world, but different places on your tooth.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.